Good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled Solving Local Lab Data Challenges, an Optimized Process for Near Real-Time Normalization. My name is Sonia Hunt and it's my pleasure to be your X-Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available to you for future streaming on xtalks.com. At this point, I'd like to thank Lab Connect, who developed the content for this presentation. Connect with Lab Connect, the preeminent provider of central laboratory support services for analytically and logistically complex studies such as immuno-oncology, cell and gene therapies, and rare and orphan diseases. They offer unique and innovative services that have been specifically designed to meet the exacting demands of today's clinical trials. Leading the evolution in central laboratory services since 2002, their services are customized to fit the unique needs of your trial. Now, it's my pleasure to introduce you to your speakers for today's event. And first, I'd like to introduce you to Danuta Kuschek, who is a Senior Director of Project Services at Lab Connect. She has extensive knowledge and experience working in academia and central laboratory industry. Her key areas of expertise include operational management and overall direction, coordination, implementation, and execution of domestic and global cross-functional or single-service projects. And your second speaker is Jim Carter, Manager of Data Management at Lab Connect. After 14 years as a quality auditor and supervisor in the auto manufacturing industry, Jim made the transition into the clinical laboratory world. His experience includes performing standard patient diagnostic assays to blood bank and specialty testing such as chemoluminescent detection methods, and in 2014, he joined Lab Connect. And now I will pass over the controls to our speaker, Jim. Jim, you may begin when you are ready.
Welcome, everybody. I want to thank you for taking time out of your day to join us in our webinar about solving local laboratory data challenges. Danuta and I are very excited about getting to present this to you today. We've been working on this project for the last several years, and we're very excited to get to see this in action at Lab Connect. What we saw a couple of years ago was we saw that there was an increase in the number of clinical trial studies in high complexity studies such as immuno-oncology, cell and gene therapy, and other rare diseases that required local laboratory utilization. And so what we tried to do was solve the problem of local laboratory data and how that data is used in the clinical trial studies from a two-pronged approach, from a project management approach and then a data management approach. And there are several challenges in handling local laboratory data because different local laboratories have different normal ranges, units, different instruments. And so there's challenges in how we utilize that data, how we get that data together in aggregated data sets.
In this webinar, an overview of a local laboratory standardization process will be given to demonstrate how those processes can be optimized. The standardization exercise described here aims to capture and report local laboratory data quickly, accurately, and cost-effectively. The procedure standardizes local lab data collection through our double data entry module into a clinical database. And then the data is pushed into a normalized table, extracted, and delivered in a unified data set.
So in today's webinar, we want to really achieve 4 different types of learning objectives. First, we want to understand the challenges of working with local laboratory data. We want to be able to identify the data process flow. Then we want to talk about what is data normalization, and then talk more about the benefits of normalization.
So what are the challenges working with a local laboratory that Jim was talking about? Those are data variability in units and reference ranges. Those are also delays and errors in data entry. And those last will detain monitoring of study progress. And this will also hold up visibility into outliers, patterns, and trends. Also, variability in units and reference ranges will affect decision-making, and that will impact delay in toxicity management and those modifications.
So as we approached how we were going to address these challenges, we wanted to make sure that this solution addressed the demands on sites and be able to reduce those demands on our sites while ensuring that the data integrity process is intact, while also making this data available in near real time. So let me draw a distinction here when we say near real time. What we're really talking about in local laboratory data is the difference between having a local laboratory data available in an aggregated data set months versus what we're pointing at, which is days. 1 to 3 days is our goal to make this data available. So this process right here really solves the challenge of being able to utilize local laboratory data long-term versus short-term. So when we say near real-time, we're really talking about in just a couple of days from the time that we receive it. So what we're doing is we're increasing our data entry speed, and through our double data entry model, we are reducing data entry errors, and we're also providing a centralized report collection place. So all of this data comes into one place, warehoused in a single place, and our double data entry model really drives the data entry speeds and the data entry errors. So the second piece of this is the data variability. We want to reduce that variability, and we do that through data standardization, that is taking all of these different data and converting them into SI units, and then normalizing that data. And we're going to talk a lot more about normalization and what that means here in just a little bit.
So let me tell you where the Lab Connect process begins. It starts with a study award. When the study is awarded to Lab Connect, we will assign project management team, and we will introduce the team to the client or sponsor. Once after that, we will review the protocol, we will review the budget and any supportive documents, and we will start creating operational specifications documents, shortly OSD. OSD is several pages document that captures every study details and guides Lab Connect how to design the study and creates the database. The OSD will go through several cycles of the review between the sponsor, client, or client and Lab Connect. Once it's approved by the sponsor, it will be utilized to set up the database and do the validation, and also to create data transfer agreement between our data management team and client data team. Once those steps are completed, the training will be performed internally, and also at this point, we are now ready to accept the data. The whole process from study award to the readiness of database, it takes about 4 to 6 weeks at Lab Connect. So typically we will designate about 2 weeks for the OSD creation, and then 2 to 4 weeks for the database build and data transfer agreement creation. So where the data collection process begins, it starts with the site, where sites will bring the subject in and perform the study activity, the study visit activity, collects the specimen. The specimen will be sent to a local laboratory, which is typically lab on site or close to the investigator site. Once the testing is completed, the results will be sent back to the sites. Usually it's a hard copy of the report. Sites will also again perform the site visit activity, will also they identify the hard copy of the report and will send the report to Lab Connect. So they could either email the report to us or fax to us. Lab Connect will receive the report to a specific study email address. Reports are received in a designated inbox, specifically created for receipt of local lab reports. And then designated team is assigned to print those reports. The reports are sent to printer and retrieved from the print queue on the printer using a PIN number. The PIN number is assigned to the employee who is printing and unique to each user. So this is our secure printing part. And then reports are printed, accessioned. So that means that each report will be assigned a unique identifier, which we call accession or requisition number, and then tracked by date and time stamp of receipt. If there is a problem with the report, a query will be issued to the site. And also, if there is a need for a translation for non-English reports, Lab Connect could utilize our translation services and translate the report, or we could utilize the pointed to us the translation company provided by the sponsor. Once the query is resolved and a lab report is translated, the reports are put out for rounds of entries. So the same report will be entered by two individual people. If there is no discrepancy between those two entries, the report will be automatically committed to the data warehouse. If there is a discrepancy, a third person, and at Lab Connect is a licensed medical technologist, will step in, resolve the query, and commit the report to the data warehouse. Typically, the process the timelines that we commit to our sponsor is 1 to 3 working days, a benchmark from the time of receipt of the report to the time when the report is available on our portal. So once the report is in the warehouse, at this point, it could be viewable through our portal at www.labconnect.com. So working for several years with this service line, we learned, what did we discover working with our sites? We learn a lot that we strictly depend on cooperation from our sides. We learn also how that all of those factors that you could see here plays very significant role in our timelines. In order for us to commit and deliver the timelines to the clients, those 1 to 3 working days benchmark, we rely on all those factors. And I will start here with the cover sheet. So we ask for 1 cover sheet per report and per subject. And the cover sheet always accompanies the report. So I would like to talk about report a little bit. So the format, quality, and timing of the report are crucial in our timelines. So we would like ideally receive the report, which is 2 3-page documents versus images of report, or versus electronic medical records that could be up to 20 pages long. So you can imagine our team members going through 20 pages of document and extract few pieces of the information, visit-specific information that we need. So also, we would suggestion from us working with the sites is that the sites submit example reports from each performing lab so any systemic issues can be addressed. Also, very important factor is the quality of the report. So we would like to receive legible report with the results and with the all information that we need, so reference ranges, units, and flags. Also, the consistency of the information on the report from the same lab sites, like ranges, units, flagging with each report. And we also timing. Timing is a huge factor in our the time frame. So we would like ideally receive the report as soon as it's available at the site. So timely delivery of report versus batch report. We're seeing our experience, we're seeing a lot of batched reports receiving at Lab Connect. We're receiving numerous reports from extended collections. We're receiving numerous reports out of order, and also receiving numerous duplicate reports. So we also learn how important is communication and consistency that I already mentioned. Consistency of the data on the cover sheet, consistency of the information on the report, and consistency between both of those. So we also, the failure in those described factors will produce queries. And what did we learn about queries? We learned how important is timely communication of the queries, and also how important is retraining for the sites with repeated issues. We also experienced how important is query management process. So we could help with this part. We could produce our metrics through the metrics. So good visibility of study progress and issue could be solved by metrics, and our double data entry tool could generate a lot of supportive metrics that clients and sites could utilize. So here is the most, on this slide, you see the most comments example of queries we're receiving from our site. So it is missing demographics, year of birth, gender, mismatched demographics, so a cover sheet doesn't not match the report, incomplete screening and subject IDs on both on the report and on the cover sheets. Dates and visit not aligned. So for example, day 5 will come to us prior to the screening visit. Reference ranges not provided, missing units, missing results, multiple visits submitted under one cover sheet, incomplete cover sheets, and also illegible reports. And on the next couple of the slides, you can see the one of example of eligible report, poor quality report. So this kind of report will automatically trigger a query to the site. On the next slide, you could see also the poor quality report, and it's with no results. This is another example that will automatically trigger a queries of those reports. So our suggestion will be to review the scan report prior to sending them to Lab Connect in order to avoid query and spending extra time on the query resolution. So in summary, our lesson learned is the importance of understanding what deliverables are needed and available in order to reduce ad hoc requests and cost, and also good study metrics that LabConnect could support clients with will improve study visibility and allow sponsors to manage queries and make study adjustment more effectively.
So when we started approaching what types of metrics that we wanted to collect, we really thought that we would spend our time more effectively if we concentrated on both site turnaround times and subject statistics. So here's an example of a report that you might see, and all of this is just mock data, but it also shows that there is some variability between sites on how quickly reports are sent to us and how much time it takes for them to gather all of their laboratory results and how they send them to us. And then we have, in that red line, you see our average business days from receipt to entry. So our benchmark is 1 to 3 days. So if we receive a batch of reports, 25 reports, let's say for site ABC 016, you can see that we have 25 reports that were sent to us from the original time of collection or day of collection of the first report all the way to the time that they actually send it to LabConnect. It was 35 days. So they collected 25 reports between the first collection and then when they sent it to us at day 35. We received those reports and then processed them in 2 business days. And so that's just an example of what this report would look like. And what this does is it really gives you good insight into site performance. Are sites holding on to reports? Are they, are they gathering them and then just not sending them to us? Or are they waiting until they get a certain number of subjects and then sending them to us after a certain number, after they hit a certain number? But this is where good communication with sites comes in. So these types of metrics right here really help you address processes at the sites, because what we're really looking for is site ABF 011, where they had 2 subjects and they sent it on day 1. So that's really what we're working for. So these types of metrics really help you get that kind of insight. So the subject turnaround stats, these are really more about collecting how many different reports do we have for these different subjects and how long is it taking for them to get from the sites to us, and then how long is it taking for us to get those in into the database. So then we wanted to look at overall project statistics. So how many total reports have we received for this project? How many are we expecting to get at different points? And are the number that we've received matching the number that we're expecting to get? And then are we missing any? And then what about sites? Have they made any available, or are they holding on to some? So will that impact how many reports that we received? So there might be a discrepancy between how many they say they've released to us and how many we've actually received, and then we have to investigate that. So cumulative reports like this, and it's important to do these frequently because you want to be able to address your problems as quickly as possible. So these types of metrics become very important to you for your study management. And so if you're interested in looking at your study progress, you would probably look at a chart like this. And because this really shows you the number of reports that you're expected to be completed in terms of a percentage. So, how are you doing? Where are you at? So these types of metrics in terms of percentage really give you an idea of where you're at from where you expect to be. So the number of the percentage of visits available, how well are we doing? Are we at 100%? And what's interesting about this one, this metric here is that it could actually exceed 100%. So if you're getting unscheduled visits, that will contribute to your count. If you're getting duplicate visits, that will also. So this really gives you visibility into what types of reports that you're actually receiving and how you can manage those. So that kind of moves us into the normalization of data in a clinical trial study. So typically, normalization is actually performed by third-party analytic groups in virtually every single study that collects data. So if you're collecting data from multiple sources, normalization is a part of how you analyze data. What we're trying to do is we're trying to move that process from a third party that could delay your visibility into trending analysis and move that into your study progress. Throughout the life of your study, you can actually see the trending of your data in near real time. So as soon as we get it committed to the database, you can actually see your data, and then you can request data transfers of normalized data. So we want to take all of this disparate data from all of these different data sources, aggregate them into a single unified data set, and allow you to be able to see your data much sooner and more effectively.
So what is local lab normalization, shortly LLN? It is the process that transforms the data from different laboratories into a set of values which are directly comparable. What normalization needs? It needs appropriate statistical models. It needs local lab results in standardized units. And also it needs a standardized laboratory, either a real laboratory or theoretical convention must be chosen. After normalization, the assays values could be treated as if they were obtained from the standard laboratory. And also normalized data can then be analyzed and visualized within a data visualization portal.
So let's talk for a minute about data modeling. So what's really important, before you can ever get your data normalized, you need to model your data. And so what we did was we took CDISC and SDTM standards, and we used those standards to create a data set. And we did that by creating two different data sets, and that is the raw data as we get it, and then standardized it to SI units, and then the normalized data. And so I'm going to walk you through these different steps that we do so that you can understand how we actually do this. This standardized data is really the first piece that you want to capture. So the data is captured through a double data entry module, and then we extract that data and we pass it through mapping tables, and we're going to walk through that here in a minute. And then we standardize it and we convert all of this data into a data structure that we define in a data transfer agreement. So in this process, we'll create two different data transfer agreements. We'll provide the data structure in the raw form or the standardized form, and then the normalized form. So we get two different products at the end of this. And you'll see more of that here in just a minute. So the raw data is converted into standardized format, CDISC, SDTM format, as well as SI units. And then we apply standard ranges. So this is part of the actual normalization process. So there are industry accepted normalization methods. And so we take those methods. We have to select a standard set of ranges to compare all of those different ranges, or to convert those different ranges into a single data set or a single range set. And at the end of this, what's really important is that every single laboratory value that's being converted here is the same statistical distance from the high and the low of the range as it was in the original laboratory report. So in the original data that the laboratory performed and sent to us and then normalized, all of those results are the same statistical distance. What this does is actually provides any flagging that's being triggered here actually gets reported the same. But it also provides all of this data now in a single range set. That's very important when you get to the trending analysis. So what we did was we took that normalization idea and we put it here into this workflow. So once data is captured through our double data entry module and committed to the database, it is now viewable on our portal. So you can access that data as soon as it's committed to the database through the portal. You can actually see it. But there's a difference between being able to see it and then being able to utilize that data in an aggregated data set. So what we do is we take an extract of all that aggregated data, we pass it through mapping tables so that it goes into CDIC's SDTM naming conventions and data structure. And then we also convert those from imperial units, if there's any imperial units, to SI units, so that now all of that laboratory data is in SI units. And so now we have a standardized data file. Then we take that standardized data file and it becomes one product. And then we take that standardized data file and then we pass it through normalization processes. So now that we have two products, we have the standardized file and the normalized file. And each of those will have a different purpose. One could be used for data cleaning. You know, the standardized file could be used for data cleaning, checking EDCs, or, you know, looking at study progress. The normalized file is appropriate for trending analysis. So let's talk a little bit about the benefits of local laboratory normalization, because, you know, we kind of really got excited because we're really trying to solve a very deep pain point that the industry has had for years over collecting local laboratory data and then getting it analyzed. What we wanted to do was, through this normalization process, it removes analytic bias. And that is the pain point. So with all of these different laboratories and everybody using different instruments, having different ranges, using different reagents, different methodologies, there's a lot of analytic bias between these laboratories. What this does is it removes that analytic bias and allows you to compare your data against itself. So each assay is as if it comes from a central laboratory, a single standard laboratory. That's important because now this data is appropriate for analytic review. So now through the process that we've adopted, you don't have to wait for third-party biostatistical groups to process data at, you know, quarterly DSMBs or, you know, different data cuts. You know, you can actually receive this data much faster and see your trending analysis much faster, which is very important. So you can actually strategically locate your data transfers of the normalized files at different places where you want to see subject trending analysis. So if you want to, you know, look at a single subject or a group of subjects at a site, you get to see this data much sooner than having to wait for it to be processed. And also, it's very important that all of this data is aggregated. So it's all of these different laboratories now reporting into a single data set. Just a hugely beneficial process. What this really does in the end is it makes your local lab data usable faster.
Some other benefits of local lab utilization and normalization include labor efficiency. So with a simplified team structure, labor use could be streamlined. Another benefit of utilization of local lab and normalization is that it removes the global logistical constraints. So top factors are here regulatory requirements, especially in global studies, sample stability, transit time, temperature excursions, and time to deal with all those mentioned pain points. Another one is a cost efficiency, so elimination of logistics overall, and utilize central location of services for data collection and data entry, and also for data modeling and data reporting. And the huge one here is also time efficiency. The timing of data availability is far more flexible here than in typical models.
So let me just close out with a case study, because we've seen a lot of different types of requests for this service, but we really tested this with a very large study that had more than 80 different sites collecting laboratory data from over 100 different laboratories. It produced, tens of thousands of reports, and we were able to take all of that data and provide it in a single aggregated data set, in a unified data set that we actually normalized. And if you can imagine how important this was to the client to be able to take all of these different local laboratories, be able to get their data into a single location, and then produce a single data set that they could use for their analytics and be able to see their trending analysis. Just a huge benefit for them. Very complicated, lots of different sites, lots of different communication. And within that case study, we actually had translation services in it too. So that added another layer of complexity on it. But it's been a hugely successful study so far. We're still working in it, but it's really proven this model out, that this is not only possible, but that it's hugely beneficial to our clients and our sponsors. So really, the whole purpose of this is to simplify and standardize our approach to gathering and reporting local laboratory data. For our clients, it becomes cost efficient. It provides better, faster visibility into their lab results, and I think you can see why. And it also provides better and faster decision making. And if you're a medical monitor, then you understand the importance of that. You understand how important it is that you can make decisions much earlier. We reduce the risks by having fewer intermediate steps. So the fewer groups that are involved in this, the lower the risk. And it also provides better control of your data. At the end of the day, that's really the purpose of that's really what you want to get to, is be able to have control of the data that you're collecting from your local laboratories.
Thank you for attending our webinar on solving local lab data challenges. We hope you enjoyed it. And now, Sonia, we can start with Q&As.
Oh, yes, we can. Thank you very much for that very insightful presentation. I hope everyone enjoyed it. And just like Danuta said, we are now going to start the Q&A portion of our webinar. I have received some questions from our members, our audience members, so I'm going to start with those, but I will encourage our audience to continue sending in their questions using the questions chat box. Okay, so Danuta or Jim, are you ready?
We are.
Okay, then. All right, here we go. Here's the first question. Is the LabConnect platform 21 CFR Part 11 compliant?
I will take this. So LabConnect, LabConnect configurable systems are robustly validated to ensure accuracy, reliability, consistent intended performance, and the ability to discern invalid or altered records per 21 CFR Part 11. So yes, we can say we are Part 11 compliant.
Okay, here's another question from our audience member. This one is, what is the frequency of the normalized data file deliveries?
Typically, the requests have been for quarterly reviews. So we typically send those on a quarterly basis. However, we have had clients want those on a monthly basis. We could do them as frequently as weekly. Generating these files and passing those through our normalization platform can be a little complicated sometimes in terms of how we review this data, but more importantly is how data is collected and sent from local laboratories. So really, that's the piece that drives the frequency, is how many reports can you collect in a week, how many can you collect in a month, and how often do you want to review your data. So those are the drivers, but typically it's quarterly, but can be as frequently as weekly.
Okay, thank you very much for that. Here's the next question I have for you both. What are the typical Lab Connect setup timelines? This audience member is asking.
So I'll take this. So we have different complexity studies and different timeline assigned to different complexity. Specifically for this service type, we would have 4 to 6 weeks from the study award to readiness of our database setup. And as I mentioned earlier in our presentation, the first 2 weeks is to develop operational specification document. And once the operation specification document is approved, we could develop and design our database, data transfer agreement, and we are ready to receive the data.
Okay, thank you very much, Danuta, for that one. Here's the next question. Is LabConnect processes compliant for GDPR and U.S. data protection regulations?
Danuta, I'd like to take this one. So I've actually worked with our Director of Regulatory Affairs very closely, mostly because it applies to the data, right? Mm-hmm, so that makes sense that I would be involved with that. Yeah. So yes, and that's the simple answer, but there's some complexities to that, and that is, how are sites providing this data in this environment where they're sending us local laboratory data? How are they sending this data to us? Because oftentimes this data is going to contain personal information, personal health information that is prohibited from being transferred. But there are data elements that can be captured, personal data elements that can be captured and then sent to us. So what we do is we work with the sites to make sure that they redact the information that cannot be sent to us, but ensure that they provide the data elements that do need to be sent to us. We work with them to ensure those requirements, and we do that within the environment of GDPR and U.S. data protection regulations.
Okay, thank you very much, Jim, for explaining that. Here's our next question. The question is, does LabConnect support translation or international reports?
Yes, we can support this request. So we could utilize our vendors for translation needs. And translation typically takes 2 to 4 business days, depends on the language. So usually it's for 2 to 3 pages reports that contains about 160 words. And it depends on the language. It could take 2 days for more common languages like Spanish or German, or up to 4 days or even 5 sometimes for less common languages.
Okay, thank you very much, Danuta. Here is our next question. Does LabConnect provide site management?
Yes and no. So
okay,
again, another complicated question. We don't directly provide site management, but what we do is we offer, very strong support with CRAs and sponsors that do have their own site management. So we provide communication on things like queries, missing reports, any kind of, data cleaning that we need to do, any communications for retraining that sites might need. So while we don't work directly with the sites, we do give them very strong support.
Okay, thank you very much, Jim. Here's our next question. Is standardized data available faster than normalized data? For example, is standardization automatic post-DDE?
Yeah, so I'll take this one. Yeah, and that's actually how the process actually works. Typically, what we've seen that the standardized file can be automated. So we can produce this in an automatic fashion to to get sent out to our clients on a regular basis. And typically our clients want to see those, monthly, sometimes weekly, depending on, what kind of data that they're trying to capture. And the standardized or the normalized file is processed out of the standardized file. So yes, it's actually a lagging file. So once the standardized file is produced, the normalized file lags behind after processing, but we can still produce it, at a much more frequent basis than you would see in a third-party analytic work.
Okay, that's interesting. Thank you very much, Jim. Here's our next question. How long does it take for LabConnect to enter reports once received from sites?
So it could take our benchmark and our timelines that we commit to our clients is 1 to 3 business days. But in order to deliver this time frame to the clients, we, as you remember from the webinar, we rely on site cooperation, and we depend on so many different factors like the report quality, cover sheet that accompany each report, the communication, the consistency, and the site retraining when we produce a lot of queries. So all those factors play a significant role in all those timelines that we really depend on. And having just to refresh everybody, the good quality report with the right format and the timing, ideally what is really important. And what triggers and creates a lot of queries is the timing of receiving the report. So ideally, we always will ask the sites and the clients if we can receive this report in almost as soon as it is available at the site. So what that means, if the report is clear, if there is receiving individual report versus batch report. So the big issue here is if there is an issue with the report, with the very first report that we receive from the site, if there is a problem, we could resolve this problem and the sites will know not to do it again. So, but if we receive a batch of 50 reports and each report will have the same issue, this means 50 queries will be introduced to the sites, and the sites will have to go through all those 50 reports. So it's always easier dealing with the 1 query versus 50 queries. And also, if we receive batch report within a month or 6 months, when you saw, as you saw on those graphs in those metrics, that means that 6 months is delay from our committed benchmark, 1 to 3 days.
All right. Thank you very much for that. It looks like this is going to be our last question. Audience members, if you do want to get your questions in, please go ahead and do so now. If not, this will be our last question. So are you ready, Danuta and Jim? And here it is. Okay, here it is. Does LabConnect support special flagging?
So, for this specific service for the double data entry normalization, the flags need to be a part of the report for us to enter. And the portal access is provided and the information for the requested user is captured in our operational specification document, but we can access the issue access to our portal at any time. So, but we could send a notification when a low, high, or critical flags were entered, so the client doesn't need to go all the time to our portal and review the raw data from the sites that we receive, but based on the notification of the flag, they could go and review that particular report.
Okay. Anything you want to comment on that, Jim?
No, thank you, Danuta.
Okay, she did it? Okay, perfect. Oh, look, we got another question that just came in from one of our audience members. Let me ask this one here. Can users pull raw or standardized data from the portal themselves? And if yes, what formats are available?
No. So on the portal is really used for, looking at study data per subject basis. So you can see each of your subjects and all of their visits, and then you can select those and expand those and see the data that way. And you can pull off individual reports from the portal, but you can't access the standardized or the normalized file that way. Those are generated through data management tools that we have. And it also gives us an opportunity, and you'll probably be glad for this, because if you could pull your own report without anybody reviewing it, you may not like what you get. So what we do is when we pull that data off, we pass it through automated QC. So we have about 75, sometimes 100 different edit checks that we pass this data through to make sure that the data file that we send conforms to the data specifications that we've defined within the DTA. And that's true for both the standardized and the normalized files.
Okay, thank you very much, Jim and Danuta. Well, it looks like that is the end of our Q&A portion of the webinar. I'd like to thank our audience members who sent in those questions. Thank you very much for doing that. If you have any further questions, please direct them to the email address that's on your screen, and there it is, info@labconnect.com. Thank you, everyone, for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Now, I'm about to send you a link in your chat box. You'll be able to review the recording of this event at that link and also share this link with your colleagues when they register for the recording as well. And we have a little bit more for you. Get even more out of today's presentation by downloading the supporting materials available under the Handouts tab. And there's 4 handouts there that you can choose from. So I encourage you to do that. Now, please join us in thanking our speakers, Danuta Kuschek, Director of Project Services, and Jim Carter, Manager of Data Management, both from Lab Connect, for that very insightful presentation. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X-Talks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Thank you very much, Danuta and Jim.
Thank you. Thank you all.
Okay, bye, everyone.
Bye.